Hi, welcome to today's mind map lesson where we're going to be reviewing all of OCR A-Level Biology Unit 2.6, Module 2.6, which is basically about the cell cycle, cell division, talking about mitosis and meiosis, and then we're also going to be looking at some specialized cell types that you need to know. So hopefully you've got your sheet ready and printed. This is the A3 version to give you an idea of what it's kind of going to look a bit like. And I've also set you this version as well, which is the A4 printing version, where you can cut out these individual pictures and arrange them in the mind map sort of orientation that you would like to. Okay, so let's get going. If you need to pause the video now, get your stuff ready, get lots of colourful pens, pencils, then you can do so. And an extra thing you might do as well before we get started is just look at all these pictures here behind me and see, what do you know about them? What features can you remember before we go into more detail? Okay, so pause the video now if you need to do that. Okay, so here we are, top left corner, and this image is really where we're going to begin. This here represents the cell cycle. Now, the cell cycle is a sequence of events that cells go through as they grow, and then they divide, and then they grow and divide, and so on and so on. So we can divide up the sequence of events that they go through into different phases. And the first key division is that we call the phase where they're actually dividing the nucleus, we call that mitosis. Too big. Let's go back. Yeah, that'll do. And we're going to look at mitosis in more detail over here. These are the subphases of mitosis. More on those later, but we call those subphases of mitosis, we call them prophase, metaphase, anaphase, and telophase. And those are all just the subphases of mitosis, so they're all happening in here. At the end of mitosis, we actually have a separate event, which is called cytokinesis. Okay, so cytokinesis is actually the division of the cell, specifically the cytoplasm. So the definition of mitosis is, I'm going to put it above here, the division of the nucleus. And I think it's important to underline that word nucleus, because this is why we say that, like, bacteria, they don't undergo mitosis because they don't have a nucleus. So this is just cells with a nucleus. Animal cells and plant cells both undergo mitosis. Eukaryotic cells undergo mitosis, not prokaryotes. Okay, so cytokinesis is the actual splitting. So really, cytokinesis kind of marks the end or the beginning of the cycle. So I'm going to draw a sort of bold line down there. That is where we start. So if you're a new cell, just been split off from your parent cell, at cytokinesis, that's where everything begins. Now we go through the major phases. G1. G1 is the first growth phase. Then we go through S phase, which is DNA replication phase or synthesis phase. And then we go G2, which is the second growth phase. So what occurs in those phases? Let's look at them. So G1. Growth phase 1. Oops, Mr. H. Growth phase 1. Then we have S phase down here. I'll change the color to kind of keep with this color scheme here. I like to think of it as synthesis phase because it's DNA synthesis. That's not the name, it's S phase, but just I think of S synthesis. So the main thing that happens in S phase is DNA is replicated. And underneath here, I've got this image from the previous MyMap, which shows semi-conservative replication. And then we have G2, keeping the color scheme that they've chosen here, is the second growth phase. Okay, so why have two different growth phases, and why is it arranged like this, and what are the differences between those phases? Okay, so first of all, let's look at G1. In G1, the cell's growing. So it's growing, making proteins, so transcription, translation is happening. Translation is happening. New proteins are being built. Also, we have replication of organelles. So that's things like mitochondria, chloroplast, if it's a plant cell, those sort of things are dividing. So the cell is just kind of growing, essentially. S phase, like we've said, that is when DNA is being replicated and semi-conservative replication is occurring. It's important that we know that by the end of S phase, each chromosome, how shall I draw this? Each chromosome is in this kind of X style of orientation. That doesn't mean we can see the chromosomes like this. I want to clarify that. We can't see that the chromosome is an X. But when a chromosome is an X shape like that, it's already been copied. So we've got one copy on one side. If we see over here, this would be one copy of the information on that chromosome. And on the other side, we've got a second copy. So by the end of S phase, we've got this kind of X. The DNA has been copied. At the beginning here, it wouldn't look like an X. It'd just be a vertical line. So let's actually show that here. So what's happening? Oops, tablet's frozen. There we go. So what's happening is we're taking one kind of copy of the DNA and turning it into that, and that is happening in S phase there. Okay, so from there to there during S phase. Okay, G2, the second growth phase. What's happening there? Well, the cell is kind of really just getting ready for mitosis. So it's kind of more specific. So the cell is producing certain proteins that are going to be needed in the mitosis process. For example, there are these enzymes, proteins called cyclins that the cell is producing lots of to get ready. So let's put e.g. cyclins. Okay, so all this process is very, very controlled, okay? It is controlled because if it's not controlled and cells divide too much, that can lead to cancer. So I suppose the first thing we should say is that one way that cells can control this process is to opt out of it. So this here, G0, what is that? Well, G0 is a kind of an optional process. Or G0. It's going to stop. So that means once a cell is in G0, it's not actively growing, and it's kind of taking time out from the cell cycle. So there's no preparation for the division of the cell happening in here. So sort of, sometimes we use this word quiescent. Basically means a bit like quiet. So the cell is quiet. Well, it may be doing things biochemically, but it's not preparing for division. Okay, so that is G0. It can also be referred to as the gap phase. The gap between cell cycles. So sometimes cells will come out of the cell cycle, sit in the gap phase for a bit, and then go back into the cell cycle. And sometimes cells will sit in the G0 gap phase for a very, very long time. So it depends on the cell type how long this is. So we could say length, question mark. And for very rapidly dividing cells like endothelial cells in the intestine. Now, in the intestine, there's food moving through your intestine all the time, and the cells kind of get brushed away, so they constantly need to be being replicated to produce more cells to replace the lining. So endothelial cells have pretty much no gap or minimal gap phase, whereas things like neurons in your brain, they, I'm afraid to say that by the time you're your age you are now, and in fact, I think when you're quite still very young, you aren't producing any more neurons. I think maybe it's like like age 5 or 6 or something. Certainly in your early childhood, you're not producing any more neurons. So your neurons are now sitting in G0 phase, and they will be doing so for the rest of your life. So protect your neurons. So neurons, could be 80 years. 80 years or so, your neurons will be sitting in their G0 phase, just sitting there. Okay, so very big differences there. Right, okay, the other thing to talk about in terms of how this is controlled is there are checkpoints, and checkpoints are extremely important, okay? So checkpoints. Now there are two very important checkpoints. There's one, you spotted it, and there's the other one down there. So this one here is called the G1S checkpoint. Okay, and that is the first major one. So between G1 and S, this is a very important checkpoint. And actually, once a cell has gone past this checkpoint and entered into the S phase to copy the DNA, it is committed to go through that cell cycle and to go on and divide. So before you want to start copying your DNA, you need to check that the DNA is not damaged. So there's a lot of checking that goes on there to make sure the cell is not damaged. To make sure the DNA is not damaged. So let's write, is the DNA damaged? That is something that the cell looks into. Is it safe to proceed, kind of? And there is a very important gene that is involved in this, which is called P53. Now, P53 is a gene which makes a protein called P53, and that protein is involved in detecting DNA damage. And this gene is actually called, it's a type of gene called a tumor suppressor. I think I just did the American spelling there, so in English it's OUR, tumor suppressor gene. And basically, tumor suppressor genes, if you have a copy of that gene and it's working well, then you're less likely, a cell is less likely to start dividing uncontrollably and potentially become a tumor. So that's why it's called a tumor suppressor gene. But sometimes a copy of this gene can be mutated and it won't work. So if a tumor suppressor gene becomes mutated through random mutation that happens in a cell, then a cell is more likely to go and become sort of dividing uncontrollably. So I like to think about tumor suppressor genes are sort of like the brakes on a car, okay? Because if they're functioning properly, they will brake and slow down and stop the cell cycle if something's going wrong. But if they're mutated and the brakes aren't working, then you can't stop and you're going to have a runaway car, i.e. a tumor. So I'm just going to put that there, brakes. Okay, there's another checkpoint up here, which is the G2M checkpoint. And in fact, there's also another checkpoint, which you don't need to know as much about. Right in the middle of mitosis is actually a metaphase over here. And there is I'll put a little bit more on another type of checkpoint up here. You can also have something called an oncogene. And these are also genes that are involved typically in checkpoints. And oncogenes, again, if they're working properly, they control the cell cycle. If they're mutated, they can cause the cell cycle to kind of run away and be uncontrolled. The difference between an oncogene and a tumor suppressor gene is we can think of, just to kind of get an idea of it, oncogenes are a bit like the accelerator, okay, of a car. So if you have broken brakes, that can lead to a runaway car. Oncogenes kind of actually push the cell cycle forward, but if the accelerator is broken and slammed to the floor and won't come up, that's kind of how we have a mutation in an oncogene. It causes runaway cell division. One thing I realized I didn't mention, hopefully you spotted it, it was more of an obvious thing, is that these phases together, G1, S, and G2, are all together, they are called interphase. Okay, so that is what this orange outer circle means. So the whole thing, everything that is not, whoops, that didn't go so well, everything that is not mitosis is called interphase. Okay, so all the way around there and all the way around there, you see where I'm going, all the way up there. Okay, so interphase includes G1, S, and G2. We put them all together, we call them interphase. Okay, so that is the cell cycle in brief. Now let's move on and talk about mitosis. Now remember, mitosis is just this part right up here. Okay, mitosis is the division of the nucleus, and it happens in a few key phases. Okay, so before we get into the different phases, prophase, metaphase, anaphase, telophase, I'm going to drop in a little video here where you can see a real microscope image of a cell going through these phases. I want you to sort of see if you can kind of identify where the cell is at what different phase. Okay, so watch that now. All right, so now let's talk about what was happening in that video. What did you see happening at every individual step? So first of all, let's label these diagrams here. Now, all this information is on your book page 148 and 149. So if you'd like to go into more detail and add more detail underneath here than I'm going to, then you certainly can. So first of all, we've got here in prophase, we've got the chromosome condensing and becoming visible. So chromosome is condensing and is present as sister chromatids because it's already been replicated at S phase. Now in the center there, the central point, if I just sort of highlight that over here, this central point of that X-shaped chromosome there is called the centromere. Okay, and that's what this is pointing to there. Just about difficult to see, that's centromere. Around the outside there, this arrow here, I'm just going to talk about it underneath, is the nuclear envelope is breaking down. So that's this one. Is sort of starting to break down. And it will be broken down by the end of prophase. Also, over here, well, there's a green one over here. I'll just kind of label it in green underneath so you can kind of keep the color scheme. The spindle is starting to form. What is the spindle, you might say? Well, it's basically microtubules made of a protein called tubulin. So sort of special arrangement of microtubules coming out of the centrioles. Okay, so then we move on to metaphase. Now, the thing that you see in metaphase is that the chromosomes are lining up down the center of the cell. We call this actually the metaphase plate. So chromosomes have attached to the spindle. So let's just kind of change color. So I'm just going to think I'll write underneath actually. So chromosomes have attached via the centromere, which is the thing in the center, to the spindle fibers. And they line up down the center. So in terms of what these labels are here, this bit here is the spindle fibers. I'll actually just let's highlight those in sort of yellow or something. So these things here, this is the spindle fibers. I'm drawing in yellow. And down the center, let's highlight in green, right there. Can you see that? Let's do it in pink, actually, it's better. That's better. Down the center there, we've got what's called the metaphase plate or the equator. So it could be called either thing, metaphase plate or the equator. Basically, it's the midline of the cell. So I will write that in a similar color to keep the color scheme. So did I say cells line up? I meant chromosomes if I did. Chromosomes line up down, sorry, let's say be specific, on the cell equator, or you could say the metaphase plate. Those are the same things. Okay. All right, moving on into anaphase. Anaphase is basically as soon as the chromosomes start to be pulled apart. So remember, the chromosomes consist of two chromatids. So imagine the centromere being this point here, right where my sort of heels of my hand are joining. So the chromosome exists like this, as an X shape, one chromatid over here, one chromatid over here. This is metaphase, they're down the middle. As soon as they pull apart, that's anaphase. And when you have to draw these diagrams, be very mindful of how it's drawn here. I'm not sure if I'm going to be able to do this with my hands. Yes, I think I can. So as soon as it's pulled apart, and then it kind of moves like this, okay? So the centromere is being pulled along the spindle fibers, okay? So we go from this to this, and then it's pulled that way. I hope you understood what I was trying to show there. But you can see it in this diagram here. So what's going on here? I'll change color again. Let's go back this time. So the sister chromatids are now separate. So therefore we don't really call them chromatids anymore. Now the sister chromatids have been separated, so they are now separate chromosomes. Pull to opposite poles. Okay, so one pole is kind of over here. This is a pole over here, and this is a pole over here. Now these poles are actually called centrioles. And those are the things that kind of organize the spindle fibers, okay? The spindle fibers kind of grow out of those centrioles, and that's where the chromosomes now are moving towards. Finally, telophase is basically when the chromosomes have got to the poles. They've made it to the poles. And we can see here that the nuclear envelope is starting to reform around those two poles. That's what these dotted lines represent. So let's kind of do that here. This is the dotted lines there. So nuclear envelope reforming. And we've got one set of chromosomes over here and one set of chromosomes over here, which is what this is labeled, two sets of chromosomes. Remember that after this process, then cytokinesis happens, which is the division of the cell itself, the cell cytoplasm itself. So actually, mitosis is just the division of the nucleus. Cytokinesis happens just after. Sometimes cytokinesis and telophase might overlap, like cytokinesis might start as telophase is just finishing, but it is a distinct event, cytokinesis, the division of the cell, not just the nucleus. Okay, I think that's everything on mitosis. One thing I need to add here, actually, what's doing the pulling, you might wonder? Well, that is motor proteins are pulling the centromeres along the spindle fibers, which is made of tubulin, essentially a microtubule. Okay, so now I'm going to put another video up. In fact, two videos, because it's really important to get an idea of this kind of sequence of events. First one is going to be another cell sort of viewed down a microscope, but with kind of superimposed over the top, there'll be colors and things, so you can kind of see what stages things are happening in, and also the stage will be written underneath. Now, when I'm showing that video, don't worry, sometimes they'll include more phases than we need to know about, like pro-metaphase is kind of like halfway between prophase and metaphase and stuff like that, or like early anaphase and late anaphase. You don't need to know much about the distinctions between those, but as we proceed in biology, we can kind of divide up those phases into even more smaller little pieces, but don't worry about that too much. Okay, so in the second video that I'm going to show you, this is taken with a fluorescence microscope, and we can see here that the tubulin proteins that make up the spindle have been labeled with a fluorescent green color, and the chromatin, so that's the DNA, the chromosomes or the chromatids, those have been labeled with a fluorescent red. So that really enables us to see how the spindle attaches to those chromosomes and then pulls them apart. So watch this. I really, really like these images taken with fluorescence microscopy. Okay, so before we move on to meiosis, a couple little things to kind of clean up, going into a little bit more detail about the applications of mitosis. So, okay, why do we have mitosis? What is it about? So mitosis, I think we can fit this in over here. So mitosis, it produces identical cells. So therefore, in large multicellular organisms, it is for growth plus repair. Okay, so if you accidentally cut your hand or something, for example, those cells have got to be replaced. So the skin cells and the cells underneath the skin will divide by mitosis and replace the cells that are there. If you're wondering about why do you get scarring, like if you to cut yourself, graze your knee, then the skin doesn't exactly heal exactly perfectly. That's just a sort of defect of the way that we grow back skin, because not all of the cell types in skin can grow back perfectly. So mitosis can also be used for reproduction, but it is with asexual reproduction in certain organisms. Okay, so that could be plants, for example. Plants can sometimes reproduce asexually by sort of growing runners underground, and the new plant pops up next to the original plant. That would be asexual reproduction. Remember, we don't say mitosis is happening in prokaryotes, because mitosis is specifically the division of the nucleus, and in prokaryotes we don't have mitosis, we have something called binary fission, okay, which is the splitting of a bacterial cell. It is worth, just whilst I remember that, where am I going to put this on my mind map? Binary fission. I'll just put it a little bit above. Note, binary fission, which is a bit similar to cytokinesis, is not equal, this is a math sign, is not equal to mitosis, because, let's do that up there, so not in prokaryotes. But we do have mitosis that occurs in certain eukaryotes that divide asexually, so we could talk about plants, Or the last one is yeast. And in year 13, I think, or is it in this year or year 13, but somewhere in the syllabus, you need to know that yeast replicates asexually by mitosis, and that process is called budding. Okay, so basically a big cell grows off of the small cell, the little cell, and then they kind of separate. That's called budding. Okay, one other thing we can do when we're looking at mitosis is something to do with something called the Mitotic Index. So that's what this picture here is. Now this is a smaller version of a larger picture that I used, and normally when we were trying to work out the mitotic index, we'd look at a larger image with more cells, but I wanted to fit it on this man map. So what you could do, if I pinch in, to work out the mitotic index of this tissue, of this cell. So these are plant cells here. So this is a oops, too large. This is a root tip squash. Okay, so we can see cells in a root tip, and it looks like a lot of them are undergoing mitosis. If I pinch in even more, which of these cells are undergoing mitosis? Which cells can you see the chromosomes? If you can see the chromosomes individually, if they're in prophase or metaphase or anaphase or even telophase, then that cell is undergoing mitosis. If you can't see the chromosomes clearly, so if you just see a nucleus but not the individual chromosomes within, then that cell is not doing mitosis and therefore it's in interphase. And the mitotic index is really just the percentage of cells that are going through mitosis. So it equals the number of cells in, and I'm actually going to write PMAT here, okay, because any of those phases, prophase, metaphase, anaphase, telophase, divided by total number of cells. So you could try and work it out for here. Why don't you pause the video, have a go. How many do you think are doing mitosis there actively and how many are not? Okay, let's do it together. See how you did. And we'll probably have slight differences of opinion here, but let's try our best. I'm going to do blue for one's mitosis. Okay. Actually, let me just write on top of it. I mean, that's going to be better. Okay, so 1, 2. Don't think so. I think that's a 3. 4. That's a tough one. Maybe that one. It's pretty small on my screen, so I'm struggling here. Maybe that's 5. I can't really tell. You might disagree. That's definitely 6. That's definitely 7. That's pretty sure that's 8. That's 9. That's definitely a metaphase. Ooh, that's a toughie. I don't know about that one. Maybe, probably just about prophase. Okay, so that's 10. So 10 cells are undergoing mitosis. So for this thing here, the MI, or the mitotic index, is equal to 10 over the total number of cells. How many are there? I don't know. I can't be bothered to count all these, to be honest, because this video is taking ages to do. But, you know, if you were to count all those cells, I don't know how many it is. Let's just do an estimate. 1, 2, 3, 4, 5, 6, 7 by 1, 2, 3, 4, 5. 7 by 5 is 35 cells. I don't know if that's correct. So 10 divided by 35, that will give our mitotic index for this thing. I can't do that in my head. So 10 divided by 35 equals 28%, 0.28, or 285, sorry, so it's 29. Or 29%, okay? So that's mitotic index. Now, why would we want to study the mitotic index? Why? Well, in humans, cells should sort of have, within a range of a set value of the cells of a mitotic index. So one application of this might be smear tests, okay? So a smear test is a type of test that women go through every few years once they reach a certain age, which looks at the cells in the lining of the cervix, and basically it's trying to detect if cells are dividing too much, because that could be a sign that there might be early stage cervical cancer. Okay, so what doctors do when they do this smear test is they take a sample of cells from the cervix, They smear it across a microscope slide, I believe, and then they look at how many cells are actively dividing and versus how many cells are not. Basically, they look at the mitotic index. And if the mitotic index is high, lots of cells are actively dividing, that indicates that the cell cycle is not being controlled and cells are dividing too much, i.e. it might be leading to cancer. So there would be certain values that they would look at. I don't have the values to hand, but I would imagine that typically it might only be something like 5 or 10% of cells would be dividing. But then if you found way higher than that, 20%, 30%, then that would be signs of potentially early stage tumor. Okay, so let's just make a note of something under that. Let's say tissues in animals should have a relatively low MI for mitotic index. If too high, could be cancerous or early stage precancerous. Okay. Now you might be asked to do this sort of calculation in an exam, just so you're aware. Okay, so we've covered cell cycle, we've covered mitosis. If you need to pause the video there, then grab a cup of tea. Might be a good idea now, because next we're going to go into meiosis, and then we're going to look at some specialized cell types. Okay, on to meiosis. So meiosis is covered by these diagrams over in this area. So first of all, let's put a little title. Kind of big. Meiosis. Okay. Now, meiosis is for different reasons than mitosis. Remember we said in mitosis, we were talking about these reasons over here. It produces identical cells for growth and repair and asexual reproduction. Now, meiosis is different. So, meiosis, it produces non-identical cells. It produces gametes for sexual reproduction. What's going on there? And these gametes, it's important to state that these gametes are haploid, okay? That means they have one set of chromosomes. So during meiosis, we reduce the number of chromosomes. In a human cell, we reduce from 46 to 23. We divide the number of chromosomes by 2. So we go from 2 copies of every chromosome to 1 copy of every chromosome. And that would be 23 in a human cell. Okay, so the next thing to know about meiosis is that we don't have to learn everything again about meiosis, because meiosis is basically mitosis happening twice with some important tweaks, okay? So it's 2 rounds of division. So we still can talk about the phases of prophase, metaphase, anaphase, and telophase, except all these phases happen twice. So we have, if we look here, prophase 1, then metaphase 1, anaphase 1, and telophase 1. Then we go into prophase 2, metaphase 2, anaphase 2, and then telophase 2. All right, I'm going to zoom in a little, and then we're going to start to talk about what these labels represent. Okay, so first thing that is really important is that in prophase 1 and then on into metaphase 1, chromosomes join up in what's called, the word is actually a bivalent, which is where 2 chromosomes pair up. And this is a pair of homologous chromosomes. So this is the pair. Okay, so this image here is kind of zoomed in on here. So this is a pair of homologous chromosomes. Okay, so in this case, let's say that the blue one is the, oh no, we've already got a color scheme here. So the red one is the paternal chromosome, and the blue one is the maternal chromosome. So let's say that these two that we see here are, let's say, chromosome 1. But you can see also this is chromosome 2 of this cell that we can see, the second set of chromosomes, and they're also pairing up in a homolog. Pairing up in a bivalent, or a pair of homologous chromosomes. So what we can see on this diagram is the labels. So here we've got the sister chromatids. So those are the chromatids of one chromosome. Trying to write this quite carefully. Sister chromatids. So this one and this one. That's one sister chromatid. That's the other sister chromatid. We also have non-sister chromatids. So these, again, over on the red side, we've got sister chromatids here of the paternal chromosome. Now, we can say that there are non-sister chromatids. So if we were to look at this red chromatid here, and then this blue chromatid here, they would be non-sister chromatids. And what happens is these non-sister chromatids, they actually wrap around each other and cross over. So that is what that is called. This is a crossing over point. And it can also be called a chiasma. So a crossing over point is a chiasma. It's a new word. That's the singular, chiasma. The plural is chiasmata. Okay, so chiasmata. And it's actually this crossing over that holds the two together in the bivalent. I don't think bivalent is something that comes up in the syllabus, but I'm just going to use it anyway because it's an easy word to say. So this is called the bivalent, but you could call it a pair of homologous chromosomes if you'd like. Okay, so that's a massive difference. In mitosis, if we look up here, the chromosomes were aligning sort of independently. Okay, they were condensing independently. We didn't have them paired up. So meiosis, we've got them paired up. They're paired up in their two chromosome 1s together or two chromosome 2s together, etc. Okay, so let's move on. In metaphase, much the same sort of thing happens. Oh, sorry, I should also say that in prophase 1, just like in mitosis, we've got the nuclear envelope is breaking down. We've got all the sort of things that happen in mitosis in prophase 1. The other things. Okay, so now metaphase. Again, it's quite similar to mitosis, except instead of individual chromosomes lining up down the metaphase plate, we've got the pairs of chromosomes lining up down the metaphase plate or the equator. So we can call this the equator. You could also call it the metaphase plate if you wanted to. This is the centriole. These are the spindle fibers. Remember, they're made of tubulin. And then we've got the centrioles at either side, which are about to pull them apart. So the key difference here is that we've got the pair of chromosomes lining up down the equator or the metaphase plate. Then, still via their centromeres, remember that's the little dot in the middle, the chromosomes are attaching to the spindle fibers. So that at anaphase, they're pulled apart. Okay, so I'm going to zoom in a little bit more for this anaphase diagram. So anaphase, again, similar to mitosis, chromosomes being pulled apart. But here's a key difference, okay? In this anaphase 1 of meiosis, it's the homologous chromosomes that are separated, okay? This one over here is largely paternal, and this is largely maternal chromosomes being separated, whereas in mitosis, it is the sister chromatids that are pulled apart. So that is a key difference. But something else is also happening. There is exchange of materials. Can you see, if you've got the color version here, that there's a tiny blue bit there and there's a tiny red bit there? This crossing over here, what's happened is we've had basically a swap. So this blue bit here maybe would break and attach to the red. So we'd have like blue and then red, and this red bit here would break and attach to blue. So we'd have mainly red and then blue. So we've had an exchange of genetic material. So let's label that. I'm going to do that in pink. because it's halfway between blue and red. So exchange of genetic material. Between non-sister chromatids, non-sister chromatids. So easy to get those words wrong, chromatid, chromosome, but you've got to be precise. Okay, so that's a massive difference as well. That doesn't happen in mitosis because there are no pairs of chromosomes. Telophase 1, pretty much as mitosis, the nuclear envelope starts to reform. We've kind of finished that first round. And once we've got to here, telophase 1, we move up to here, to prophase 2. I'll zoom out a teeny bit. Oh, that's too much. There we go. So once we've gone from telophase 1, we go up to prophase 2. So we go prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 2, metaphase 2, anaphase 2, telophase 2, like that. So now we're up here at prophase 2. So the second round of meiosis, the second division of meiosis, is more similar to mitosis. So we've got the centrioles, which have already been used in the first division. They have to replicate and move to the poles again. Because they're going to be used again to divide the second time. And we've got new spindle fibers. Now they form at right angles to the direction of the original division. So basically, I'm going to put a sub note under here. So the first time round, the division is happening kind of like along this axis. Okay, can we see that here? It's divided that way. But then the second time round, in the second division, the division is going to happen at right angles to that. So it's going to be, the division is going to go up, down. So we can see that here. So new spindle fibers form at right angles typically. Okay, metaphase 2. Here we have chromosomes on the equator. So this is very similar. In fact, it is the same as what happens in mitosis. The chromosome lines up on the equator. Notice though that some of those chromosomes, they're not all one color because we've had crossing over. So each of those chromosomes is subtly different. Here we have anaphase, so now we have the chromatids are being pulled to the poles. And then we've got 4 haploid gametes produced. Okay, so thinking about this sort of haploid gametes idea and the difference between haploid and diploid, there is a classic question that can be asked about this. And I've seen it come up a fair few times. So it is a kind of questions about this sort of graph. So we can draw a graph of the amount of DNA measured in arbitrary units over time in a cell that's going through meiosis. So this is amount of DNA brackets arbitrary units, which means just they kind of made them up. And this is time. Okay, so cells in mammals, diploid cells, they start out with kind of an amount of DNA as 2. That's because there's 2 of each chromosome. We start out with 2. So now we're in a sort of G1 phase of the cell cycle, and then we enter S phase, and the DNA is copied. Okay, DNA gets copied and copied and copied until there's 4 sets of DNA. Okay, and then we go through. This would be, so I'm just going to label this. This would be S. This would be G1. This would be G2, and then we go into meiosis. Instead of going into mitosis, we're going to go into meiosis now. But the first mitotic division would happen, and basically, suddenly, whoops, that's supposed to be a vertical line, we have the first division of meiosis, so that would be M1. There might be an ever so small gap, tiny gap really, and then we go into the second division of meiosis, and then we go down to here, okay? So then we'd end up, this is supposed to be 1, with only one copy of the DNA. So basically what I'm trying to say is that at 2 here, this level, this is diploid. Tablet being weird, and down here this is haploid. Okay, so we have synthesis phase, bit of G2. This would be the first division, so meiosis 1, and this would be here. This would be the second division here, meiosis 2. So just be aware of that sort of graph. Okay, right. I'm going to zoom out, and actually I'm going to put this over to you now, because I spent quite a long time doing this video, mainly because I was trying to figure out how to embed other videos into it, which I hope you enjoyed the animations. And now what remains for you to do is to make some notes on specialized types of cell. I'll quickly go over them for you. I'm not even going to write anything. So we've got guard cells here. Remember, they're specialized in how they can open and close in response to water stress. We've got here, this is an organ. This is the leaf. Remember, the leaf is an organ. All right, I'll just write a little bit. Oops. And let's make that a bit bigger. Okay, so leaf, cross-section. We sometimes write XS for cross-section. So this is the top, this is the bottom. Label all these different parts. This is a root hair cell. Label these different parts and say how they are specialized. Specifically, I want you to really focus on, in this one here, you need to focus on this layer here, which I'm just highlighting here, because that is the palisade mesophyll cells, and they are the most adaptive for photosynthesis. You can find those kind of in that information on page 155 and 156 of your textbook. I'll just write that here for you, 155 plus 156. You don't need to write this on your mind map, but there's the information. That's where you need to get it. And then we're going to do some work on animal tissues later on, not in this mind map, but in terms of animal cells, you need to know what these different cells are in a blood smear, and you'll find that on page, where is that? Oh yeah, page 153 of your textbook. This also comes up in disease, which we're going to be looking at later, and the immune response. And then here, this is the sperm cell. Now, the sperm cell sort of is linked to meiosis because it is a gamete. Okay, so a gamete could be a sperm cell, which contains varied. A gamete all the gametes are different from other gametes. So all the sperm cells that a person produces, or all the egg cells that a person produces, are different from each other because they have been produced through meiosis, which generates variation. Now, just I'm going to highlight, before I let you do this on your own, 2 key sources of variation. One is crossing over. That is a source of variation. I've highlighted that in yellow. And the other is actually at anaphase/metaphase. I'll label it. We've got independent assortment. I think that is key. Independent assortment. Okay, so that means, and if I pinch in a little bit, you can see that. That means that here, let's say the red is the paternal. The paternal chromosome here is going left. And underneath the paternal chromosome looks like it's also going left, but it doesn't have to be that way. Each chromosome individually decides which way it's going to go. So maybe at the top, the paternal chromosome goes this way, and underneath the paternal chromosome this way. And the next one down, because we'd have 23 of these pairs in a human cell, they go just randomly which way the paternal and maternal would go. That actually gives, if you do the maths of it, it gives, just from independent assortment alone, I won't go into full detail here, but it gives 2 to the 23 different possible combinations of maternal and paternal chromosomes from meiosis. Isn't that amazing? So you, if you're female, can produce 2 to the 23 different egg cells, and if you're male, you can produce 2 to the 23 different sperm cells. And that's not even including the variation which comes from chiasma. So in reality, it's even more. Okay, so I'm going to leave it there. We've talked about cell cycle. We've talked about meiosis and mitosis. Now it's over to you. Can you add some information about those specialized cell types? God cells, plant, leaf cells, palisade mesophyll especially, root hair cells, the blood cells, and the sperm cell. Then send a picture to me on Teams. Hope that's been useful. Thank you very much. Bye-bye.